About cell structure and the function of these cells. So characterization of living cells. They need to grow. You know, everything increases in size one way or another. You know, we did not all start out this size. We were little single-celled organisms. They need to reproduce. If you don't pass on your genetics, then your species is dead, and that's not very good for life. Responsiveness. They need to be able to react with their environment. If we put our hand on the stove and it's hot and burning us, and we aren't smart enough or can realize and react to that, we're not going to survive very well. So even down at microscopic levels, they have to be able to move towards food or away from some chemicals that are harmful. So you have to have that responsiveness. And metabolism. Everything likes to go towards chaos. You need to consume a lot of energy to keep everything stable and in its place. This is where our metabolism comes in. So that's controlled chemical reactions in living cells. Okay. So we're looking at E. coli versus paramecium. So, coli, prokaryotic, eukaryotic? Prokaryotic. Bacteria. Paramecium. Single-cell eukaryote. One good clue, look at the size. 1 micrometer, 40 micrometers. So right there, you can start going, oh, that's a lot bigger. So what is some other examples of a prokaryote? We have E. coli. Anyone think of anything? What are the two basic types of cells that fall into prokaryotes? Just talked about them.
Bacteria and archaea.
There you go, bacteria and archaea. Scientifically, we use a lot of these organisms because that's where we get TAC, so DNA polymerase, that we use to replicate DNA. We actually have gotten TAC, which is from Thermus aquaticus, which is from a hot spring. And so it's a DNA polymerase that's used to hot temperatures, so when you boil it or nearly boil it, it doesn't fall apart. So it's a stable protein. So there's a lot of them in science. Okay. And then, what are some eukaryotes besides the little paramecium? This should be super easy. Yeah.
Practically any cell in your body. Practically any cell in the human body would be eukaryotic. Other than
Animal cells?
Other than red blood cells.
Well, but still a eukaryotic cell.
No nucleus.
It had a nucleus at one point.
That's true.
And it doesn't live very long once it gets rid of it, so it has no nucleus at all versus no membrane back, so we are eukaryotes. Everything multicellular that we've come across is eukaryotic. So, knowing that everything multicellular we've come across is eukaryotic, what's another example besides animals? I still couldn't hear. Fungus? Yeah. Plants. Plants are a very common one. So yeah, algae, fungi, protozoas, these are all eukaryotic organisms. Prokaryotic cell. They have a very complex cell wall surrounding the cell membrane. So, we're going to talk about this a lot more because it's incredibly important. Their DNA is usually just a large circular chromosome, and it forms into an aggregate called a nucleoid. Now, their major source of DNA, their major DNA, is this large circular chromosome. They also have these little tiny pieces which are known as plasmids. So, size-wise, if you're looking at the difference of, here's their circular DNA, and then you get these little tiny plasmids. And they can have hundreds of them. Those plasmids are what can carry antibiotic resistance genes. So those will come up later when we talk about Hillman. Organelles. Generally, prokaryotes lack organelles. They lack membrane-bound structures. So they have, they have ribosomes and stuff, so they can make proteins, but in general, they have no real inner membrane-bound structures. They're very small, so you get they're about 1 micrometer in diameter. So again, very small, very simple. Okay. There are 3 basic types, but 2 that are very common and you should know: spherical and rod-shaped. So spherical is cockeye, and rod-shaped is bacilli. Okay. So circle, rod. So the spherical, you'll start noticing them in things like staphylococcus. So right there in the name, it tells you what shape that bacteria is. So there's multiple, so if you see bacilli, you know it's rod-shaped. If you see cocci, you know it's spherical. They can come singly, they can make chains, they can make little cuboid packs, or you can have the long chains for the bacilli. They come in multiple arrangements. Questions? Okay. So this cell wall, outside of their cell wall, they have the glycosylates. So a polysaccharide containing material lying outside of the cell wall. That sounds familiar. So they have either capsules or slime layers, and the entire point is some form of protection. Okay, so they want to be protected from the host immune system, because if they're getting in, they kind of want to stay there. They don't want to be eaten. Or they want to be able to prevent dehydration or adhere to something. And so, capsule slime layer. So pictures of these. We have a capsule, we have a slime layer. So the capsules are composed of organized repeating units of organic chemicals. They're firmly attached to that cell wall or cell surface. And it's really thought that their main function is to prevent the bacteria from being recognized by their host. Versus the slime layer, which is loose around the cell membrane, cell wall, and is very water soluble. So this means that it doesn't have a problem interacting with the water, but it's this sticky layer which allows these prokaryotes to all attach to surfaces. Flagella. We talked about flagella briefly, as far as that it's used for motility. And taxus. What is taxus? You might recognize it more as chemotaxis. So being able to know where you want to go, moving towards, away. So flagella, composed of 3 parts. You have a filament, a hook, and a basal body. And this is all made of protein subunits. And the arrangement of flagella can differ based on the different bacteria. And you can also, to make things even more complicated, you can have spiral bacteria, which has endoflagella. So right there, endo. Anyone know what this is? Inside. So another thing with science, learning roots helps, because then you can make up or guess and then see how right you are. So endoflagella. So they have their flagella actually on the inside. It's wrapped underneath the cell's outer membrane, and it helps them they actually end up spiraling. Okay. Flagella are not present in all bacteria, but they're present in a lot of them. When we talk about bacteria, we're going to mostly usually end up talking about gram-positive or gram-negative. It's one of the most basic stains. We'll get into that in a minute. But you can see the 2 different structures. We have gram-positive piece of bacteria, so you have a cytoplasmic membrane, big peptoglycan layer, cell wall. You have your hook. Okay. So the difference between a gram-positive and gram-negative is how big this layer is. As you can see with the gram-negative, it's very small. that they all have a basal body, a hook. So we'll get in that a little more. So we're looking at prokaryotic cells. So this is kind of nice because they give you half of what a real picture would look like and half cartoonized, because cartoons are so much easier to understand than sometimes the real pictures. But, okay, so we go through a prokaryotic cell. This means we're going to have the nucleoid. What is that? DNA. Let's talk about, that's where it gets condensed down into that nucleoid. We have cytoplasm. Cytoplasm is just that membrane, that fluid outside, just water-like fluid. You need ribosomes. What do ribosomes do?
They make RNA or DNA? Proteins.
Proteins. Okay, so what is the central dogma of life? DNA goes to RNA, goes to a protein. Most things do this. Can you think of something that doesn't? Something we... Viruses, but only specific ones. Retroviruses like HIV, they actually have RNA genome and then they go backwards and then go through. So DNA to RNA is called, what is that process? Transcription. And then translation. You are transcribing the DNA into just a slightly different code, so you're just copying it basically. Translation, protein, amino acids, completely different language than the DNA RNA language, so transcription, translation. Ribosomes, they're important for translation. Okay. We have cell wall and a cell membrane. So cell membrane's on the inside, cell wall is protective coating. Here we have our nice little flagellum. And a lot of times you're going to have inclusions. Things just get shoved in between. Also, this is a great little picture because it also has glycoxylase on the outside. 3 basic types of arrangements for flagellum. Okay. So you can have periticores, top one, A. So that's where the flagellums are everywhere. Just cover the surface. So peri, around. You can have polar, so just one, so single polar. Or you can also have tuft of polar. Cover the surface, have only one on the end, or have a bunch on the end. And then that other one we talked about, if you have an endoflagellum, you can rotate. And so what happens is they actually get rotation inside, which makes it so that it spirals through the medium. So that's how it moves, is with a corkscrew motion. One thing that's cool with flagella is that they can, a lot of times, do what's called tumble and run. So, chemotaxis, so movement towards a chemical attractant or away from a chemical repellent. So with this one of the differences between, the difference between prokaryotic and, bacteria and archaea is whether or not they will. So with bacteria, if they're going one direction, they'll start doing a run. If they spin all their tails opposite, they'll do it separately from each other. They'll kind of tumble. And then they'll whip all the flagellum together again for a run. And so it's not organized. They don't really get to have much of a say, but the idea is, if going straight, isn't getting you towards or away from whatever you want, let's randomly try and move, and maybe this new direction will be better. And so they can cover a lot of ground this way, rather than just randomly going, it's that direction. Flagella are kind of cool because they can move 60 cell lengths per second. And so the equivalence of that would be a car traveling at 670 miles per hour. So these suckers can move. So that's why when you start looking at things today in lab, we have some chemicals to help slow them down. So that way you don't just start getting them focused and having them run off the side. So they can work at more than 100,000 RPM. And the rotation propels them through the environment. It is reversible. It can go clockwise, counterclockwise. They are capable of moving both ways. But remember taxis? There's stimuli. So, flagellum, really big, long. You get shorter ones, pili and fimbria. So, fimbria, they're thread-like projections made of protein subunits, okay, versus pili, which are for conjugation. And so, with the fimbria, their whole job, there's usually quite a bit of them. They're outside of the bacteria. They're not as long as the flagellum, but they're there to kind of help stick and make it so that things stay together. Pili also is called conjugation pili, and that's involved in bacterial mating. So we talked about bacteria, they're asexual. They do not, sexually reproduce. So then you get to that question of how is it then that bacteria start picking up immunities from everybody else, or how can you get, this bacteria now is resistant to this antibiotic, and what it is it's these pili and those little tiny plasmids. So those little plasmids can carry resistance genes. They can create these little pili, and basically take that over to another one and kind of share plasmids with the next one. And so that's their form of mating. So then the fimbria, it's all about attachment. Trying to get those bacteria to adhere to surfaces. Very important for pathogenicity. So it wouldn't work very good if these bacteria couldn't adhere to the cells they're trying to infect or invade. So an example is the E. coli being able to attack to the urogenital tract, so that way you can actually produce problems. Okay. Just a pretty little picture so you can actually see some of the size difference. We're looking at bacteria. You look at the really long flagella. So now what variety, there's 3 different basic varieties of flagella. What would you call this one? So I would say, since it's all over, probably the Perry's chorus.
Yeah.
Or you could also probably find reason to think of some of it's all polar, depending on maybe not just a bent over. So maybe it's a tough polar. Okay. But just so you can see the size difference, and it's those fimbria that are helping 2 bacteria stay together. The conjugation process. So this is what happens. This is bacteria sex. They have that conjugation pilus. That's what is one bacteria is transferring to the other. They even have a special little factor so that way only one of them is transferring their information. Pili are actually a special type of fimbria. They can be longer than other fimbria, but they are shorter than the flagella, and so it's one of those things where they can be long because they're as long as they need to reach the next one. Typically, a cell will only have one or two of these, it's not really one of their major things they need. I mean, you need to be able to grab hold and hold on to things. You don't really need to have a bunch of ways of mating. But this is how they mate, this is how they transfer DNA, this is how they get resistant to things. The other thing bacteria love to do is make biofilms. Okay? So biofilms are a bacteria community that attach to a surface. So that glycosylase and the timbria, these can be really bad because they can be resistant to disinfectants and some antibiotics. And there are 5 basic stages: initial attachment, and then they go through irreversible attachment, maturation, maturation 2, and then dispersion, so they can start it all over again. And so a biofilm is any group of microorganisms. So they're just cells that stick to each other and on a surface. They're frequently embedded within a self-producing matrix. So they create that matrix for themselves because they want to stick, they want to stay, they want a nice little home. There are also, a lot of times you might hear them referred to as slime, but they generally, they have their extracellular matrix has some DNA, it has some proteins, it has polysaccharides. They make this nice little comfortable home.
The biofilm is known as slime?
This is biofilms, yeah. So biofilms form on living or non-living surfaces. One of the really prevalent things is you find them in natural, industrial, and hospital settings. So this is where you don't really want microorganisms attaching, growing, and protecting themselves so you can't get rid of them. Okay. So, bacterial cell wall. So we've been talking about, it's on the outside, digging a little deeper into it. So the cell wall provides the structure and shape, it helps protect the cells, it assists in cell attaching, assists in, it's a nice big thick coat, so it makes it so that way antibiotics can't always, or antimicrobial drugs can't get to it. But also because bacteria have a cell wall and we don't, certain antibiotics, we can actually target that cell wall. It's one of those ways where that's why we can give it to people, because antibiotic, anti-life. But if it specifically targets a cell wall, we don't have to worry about that. We don't have cell walls, so it's only going to target bacteria. Now, it'll also target the good bacteria that we have that helps digest and give us all sorts of nutrients, but it'll also get rid of all the bad stuff. It gives bacteria cells their shape. The big thing is it's composed of peptoglycans. So this is where we get those 2 basic definitions. One of the first basic definitions is it gram-positive or is it gram-negative? So when we're talking about gram-negative and gram-positive, this is what we see. Okay? Purple or pink. So the gram stain was developed using Knox technique, which we talked about last time. And so it's a series of dyes. And we found that some microbes stayed purple, some microbes went pink. So this is where if it's purple, it's gram-positive. If it's pink, it's gram-negative. Okay? And so it just was one of those easy ways to start separating them. It's one of those first separations of bacteria. So what it really comes down to is that cell wall structure. So gram-positive, they have that thick glycosylase layer versus the gram-negative, which has that really thin layer. Okay? So this is what we're looking at. Gram-positive, you have that peptoglycan layer, which within it you have lipotetroic acid, which is important because that helps to negatively charge and give the surface a negative charge for these gram-positive cells. And because it has such a thick wall of that peptoglycan, it retains that crystal dye. So the crystal violet dye, which is what they use for the gram staining, which is why these appear purple. And up to 60% of the mycolic acid in acid-fast bacteria help the cells survive desiccation. So the idea is that because there's so much of this acid in there, it helps prevent them from drying out. Right? So, gram-positive, big peptoglycan layer. Make sense? Versus gram-negative, small peptoglycan layer. Talking right here. So they have, so for gram-negative, that has only a thin layer of the peptoglycans. It's bilayer membrane outside of the peptoglycans, and so this is why you can see, so, membranes, you have our lipid bilayer, you have the peptoglycan, then you have another layer.
Mm-hmm.
One of the important things to know, and why it's so important to separate between the gram-negative and gram-positives, is this LPS, okay? The lipopolysaccharide. And so this lipopolysaccharide can be very detrimental to us if we get an accumulation of it. So if somebody has bacterial infection, you want to know, is it gram-positive, gram-negative, so you can start to actually weed out what is the best way to treat them, what do we need to be worried about. So if they're gram-negative, what color are they? Miss Staining?
Gram-negative.
Pink. So, we keep talking about this peptoglycan layer. What does it look like? So it's a sugar backbone, NAG and NAM. So it's a tetrapeptide, 4 amino acid chains linked together. So you end up getting this structure. It's a very simplified version of it. A better look at this one. So you get the structure of the peptoglycan. So you can see that you just get rows upon rows of alternating sugars. And your book has a great little picture that shows you the difference. I mean, these are basically, you just take a glucose molecule and you add, add either an acetyl chain to it or the acetylmurate chain. So they're basically glucose molecules. They're modified glucose. And then they have these nice little tetrapeptide amino acid cross bridges. So tetra means what?
4.
4. Peptide. Amino acids, peptides. So 4 basically amino acids. So that's why you have this 4 peptide amino acid cross bridge. Does that make sense?
Yes.
So with this very sturdy structure, you start to have this, I mean, you can understand how this would be able to stand a lot of pressure and stress, and so how this can protect those bacteria. Now we talked about the LPS a little bit. Here's why it's so important. So, the lipopolysaccharide, LPS. This lipopolysaccharide, it's highly variable in its structure, can change a lot. And the core polysaccharides is very similar in structure to bacterial genes. The big thing is this lipid A, okay? So you have this part of the lipid A, you have polysaccharide, core polysaccharide. Okay. Now, this lipid A though, it can cause an accumulation of this, it can cause endotoxin. So endo, within, toxin. So you can get fever, you can get blood vessel dilation, shock, so blood clotting, overall, really not a good thing, all due to accumulation of that lipid A. Again, most bacteria the vast majority of bacteria have cell walls. There are some very few. Mycoplasma pneumonia, one example. They're very rare. Okay. So this is one of those, and the lipid A in the gram negative is one of the issues.